did you do instead? What did you do to get power, or to entertain yourself, or although I think most, because a lot of you guys were, you know, texting me through Remind, and it seemed like most everybody I talked to had power. I think we were one of the people that didn't. I felt like got hit harder. I don't know, but I obviously didn't talk to everybody. But I talked to at least 25, 30 people in our class, in the junior class, and I don't know of anybody that was out of power more than a day. I think you said you were 2 days. You're the most I've heard of.
The worst that I heard was Lily Lynn. She didn't have power the entire time.
Really? She sent me an email. I don't know how she did that. She probably had her computer charged. I guess so. Well, she had attachments of her extended essay and stuff.
Yeah, you can do that on your phone.
Okay. Hey, what's up, Monis? I'm just gonna start recording. Time I'm coming to celebrate it. Oh yeah. I don't know what Twitch is. What do you mean?
Someone said you're a streamer.
Don't know about Twitch. Santos. Oh, Santos. Diana Santos. Oh my gosh, Diana Santos. I've got a great picture of you at home. It's you. I got a picture of you with Elosa and Sarah Marino. It's actually
a picture? How?
Yeah. Hold on a second.
Oh no.
I love it. It's on this thumb drive. Yes. It's on this thumb drive. I had to get a thumb drive to print. Oh, which brings me to the, yeah, I need to pass out some work here. I came up here and printed the handouts that we needed for today and for the schools, the kids that are in class. Anyway, I couldn't find a thumb drive. You have to have a thumb drive to print here for whatever reason. The computer won't communicate with the printer. And on this thumb drive, there's a picture of you, Alosa, and Sarah Marino, and you're about, I would say, 12 years old. I bet you're 12. Maybe you're 13 years old. You guys are together. And I was like, who is this? I reckon that's Santos right there. Because there's lots of stuff on this thumb drive, I think, from the yearbook.
Where did the picture even come from? Oh
no. Yeah. You're with the teacher. Hold on, I'll be right back. I'm going to pass these things out. She's a purple. You need to get a pencil. And that phone, you're gonna have to take it to it somewhere. Yeah, we gotta get busy today. Oh, my back. Yeah, you guys were cute. I swear that you guys were 12 years old. You couldn't have been older than that.
Because I must have been back in middle school then. Ew, no.
Yeah, oh yeah, it was definitely middle school. You guys are wearing red shirts, so that's sixth grade. Or seventh grade, yeah. So you were wearing red shirts, so seventh grade, whatever age that is, right? 13. There's Mia Wallace.
Oh.
Mia Wallace. Wonderful child she is. Mia Wallace.
Yes.
Wonderful child she is. Sing it, Mia.
I finally made it.
I know it. Sometimes in the morning, it's tough to get out of bed, right? You don't have any water. Are you still without water?
It's so basically it was like, don't drink the water. And I gotta use water to get ready in the morning. So I had to use water bottles.
No, I
Which
think
is
you can
hard.
drink the water in Arlington now.
I live in Arlington.
Where do you live?
I live in Fort Worth.
And they still have a boil water?
I think like yesterday it wasn't okay, but now they're saying it is okay. But me and my dad are paranoid. So we still don't want to use it just yet. Especially since our neighbors' pipes are busted also. So
Woo. All right. Who is Daniel Hernandez? Yeah. Oh, he did? I'm going to admit this kid. I can always remove him if I need to. Let's see who this is. Hey, Daniel Hernandez, you need to identify yourself. I think you're using somebody else's account. So Daniel Hernandez, I need you to change your name to what it actually is. If you're a student of ours, I'm going to give you 30 seconds and then I'm going to remove you if you don't identify yourself. So Daniel, I need you to identify yourself. 30 seconds, buddy. You may be at somebody else's house, and that's why I normally wouldn't let you in, but I have a feeling that you may be somebody else. Oh, here we go. Here we go. Oh, Diego, good. Okay, gotcha. Gotcha. Oh, I don't know. Can you guys tell Diego how to change his name, baby? Yeah. What? Come on the mic and tell him.
And go over your name, like your mouse over your name, and at the top right, there's 3 little buttons. Click it, and it should say rename.
There you go. The three little dots, you click on that. Okay. Yeah, 3 little dots. Click on that and it'll let you change your name. Class starts at 8:30, correct? That's good. So let's say I've got 1 through 4 here. So 2 of you guys are going to be responsible for 1, 2, 3, or 4. So out of the 5 of you guys, 2 are for sure. 1, 2, 3, and 4. So you guys openly discuss these on the do now. 2 at home and 2 in class. 2 at home and 2 in class. Good morning. You guys talk it out. Talk it out. Yeah, you guys talk it out. I'll mute myself so that people can't. Well, I don't care. I don't think they can. They can hear your discussion. Be like a class discussion would be better. So 2 of the do nows are going to go to. Oh, please don't put the answer in the chat box openly. Please don't put the answer in the chat box openly. Hey, good morning to you. Oh, Diego is back. Hey, what's up? Say hello to our illustrious, beautiful, vaccinated Mr. Lenager.
Say hello to my second shot. Hello.
Huh?
Are you starting already?
Well, I mean, class starts in 5 minutes. Yeah. I mean, I just kept my advisory open. You just go right into it. Keep the kids that are already in there, right? Hey, man, are you off this period?
No.
All right, I'm gonna go re-up my coffee. I'll be back in 2 minutes. Hey, do me a favor. When you guys see somebody pop into the room, just go up here and just click and let them in, all right? All right, actually, I'm gonna make you one of you guys co-host. Let me see if I can do that. Let's see, Daniel result. I don't know if I can do it, actually. Nope, it won't let me. I don't know why it does that. All right, you guys are watching that. Daniel.
I went to Mr. Cook's executive lesson because I forgot like, the data sources. Here you go. Thymine, guanine. This should be on the website. So the Duracells. I already know on this. I just forgot the names. Cytosine. Oh.
Anybody come in? Who? Okay. Have you guys figured out all of them so far? So you figured out one of them was a phosphate group. Which one did you figure out was a phosphate group? 1, 2, 3, or 4? Which one?
4. No. Yeah.
Oh! What happened to my thing? Oh, there it is.
No. Phosphate group is one that's on the end. Ends. Okay. That's 3. That's 3. Phosphate group is 3. Yeah. I'm sorry.
Oh no! Oh no!
I pressed enter because I wanted to,
I'm sorry.
start another sentence.
Well, let's see. What about that? You guys may or may not have got one of them. You still got to get one more. And so those of you guys that are at home, 2 of these, 1, 2, 3, or 4, are going to go to one of the 5 students that are in this room. The other 2 are going to go to you guys. I need you guys to dig deep, dig deep. Let's get this day rolling. Pull on this prior knowledge so that we can jumpstart this lesson. And don't answer out loud in the chat or out to the public. I say out loud. Keep it private. Keep it private. So, Monis, you're pretty close. I got yours. B, you need to be more specific on 4, and you need to be more specific on 2. All right, now we have, yeah, I was gonna say, you need to really be careful there, Keaton. All right, now we have 3 minutes. 3 minutes, we'll start taking answers. Let's go. That will be 8:32. We'll start taking answers at 8:32. Make sure you guys are talking to each other. Make sure you guys are talking to each other. So like if y'all know something that they don't know, and if y'all know something they don't know, share that information across the class. I don't care if the kids can hear you guys discussing. Two more minutes. Let these kids in, let these kids in, let these kids in the room. I see you guys answering. I'm taking attendance right now. I won't be able to come back to you for about 1.5 minutes. There's Dev Patel and Miriam Yang. One more time. Can y'all believe that we had almost 100% attendance? (8:31). We'll start taking answers at 8:32. Oh my gosh, why is it doing this to me? Stop doing this to me, man. Ugh, there it is. Let's see. And I'm going to call on people individually. And since I have not heard you guys saying much, I've heard these two guys talking. I'm telling you, it's going to be one of the three of y'all for sure, because y'all have got to talk to each other. I heard these guys talking about it. Ooh, what was that? Okay, 8:32. Randy, you have number one. Yeah, just come on your mic. All right, that's fine. What did you get in the chat? Okay, good, Randy. And a lot of you guys were saying bases, but you're correct in saying that it's a very specific base, and specific is going to be important. We want to make sure that we are saying which base it is, and the first base is correct. It is going to be cytosine. Cytosine would be the base. G pairs with C. If I can get that sucker to the right. There we go. Excellent. Number two, that one is going to go to Abel. So what is number two, Abel? I'm sorry? I can't hear you, buddy. It is not, it's a bond. What kind of bond is it? No, hydrogen, these are hydrogen bonds. Oh, did you? Okay, well, we'll come back to that one. So if you, he says he got 3 and 4, which do you want, 3 or 4? You want 4. Okay, Abel is going to give us 4. What is this one, Abel? Deoxyribose. You are correct, sir. Deoxyribose. And that's not supposed to be an I there. I'm sorry. It is deoxyribose. Scratch out that I. All right, so let's see. Daniel Rosales, what is this? Number 3. I'm sorry. It is a phosphate. You are correct, sir. This is my phosphate group. I'm feeling good about this so far. It's supposed to be 2 words, phosphate group. And number 3, let's go to somebody at home. Let's go to, let's see, I got to see who's over here. Oh my God, why does this keep happening to me? I'm hurting myself. Let's go to Diana Santos. What is number 2, Diana? What the heck?
I said a covalent bond.
Covalent bond is correct. It is a covalent bond. Covalent bond. All right, so not Ron, but bond. So which are stronger, covalent bonds or hydrogen bonds, Diana?
Covalent bonds.
Yep. So this backbone here, this is the backbone of DNA. This is going to be very strong. These bonds here are hydrogen bonds. They're going to be very weak. There's 3 hydrogen bonds between guanine and cytosine. Adenine and thymine have 2 hydrogen bonds. You can see there's 2. And then you can just make up whatever these are. Let's call this cytosine and this guanine. I know they have to be cytosine or guanine because there's 3 hydrogen bonds. It doesn't give me any indication as to which one it is. Now, if you were taking a test, there's some of the command terms that I cannot give you guys. This would have been the command term that I would have given you if you were in class. So today you're going to have homework. I know you're like, yeah, it's what I wanted. Well, happy birthday. You do have homework. I think it was your birthday last week, right, Santos?
Yes, it was Friday.
It was Friday. Happy birthday, kid. Well, for
Thank
your
you.
birthday, Diana Santos gets homework. Diana gets homework. Yay, for her birthday. You're 17 years old, right?
Yes.
Oh, awesome. Well, congratulations. You get homework for that. You would have had to draw that. So it's not enough that you guys could just look at a figure. You're going to have to draw this figure, which kind of gets back to our project that we have. The quarter project, I want to take you guys to Schoology. It's where we all want to be. I want to go to Schoology, Mr. Pope. Please take me to Schoology, please. Jesus, I want to go to Schoology. So go to Schoology real quick. Let's go to genetics. I have played with your dates. I had to play with the dates a little bit because we missed last week. So I do want you guys to see that your quarter projects are now not going to be due on the 1st of March. They're now due on the 5th of March. Also, this asynchronous lesson, if you have watched the asynchronous lesson, you'll see that I posted a video for you guys, and that video was my version of this. I made my own project. Some of you guys say, I think it was Alan that said, could you give us an example? And I did. It didn't take me but about an hour to do it. So some of you guys have replied to me and some of you guys haven't. I know that there are 49 people that watched the video, only 49 of you. So that tells me that there are 14 that have not at least. Some people may have watched it more than once. You need to go watch this lesson so that you can see an example of what my virtual model is like. If you're an in-person, you do not have to make a hard model. Yeah, so if you guys are, meeting here, if you want to make a virtual model, you can make a virtual model. There were several people that said it's not fair, and I agree. After doing this and seeing how easy it was to do online, you could do that. Also, the asynchronous lesson exit ticket. Some of you guys did do this. Some of you guys did not do this. You need to do this. It's the easiest grade in the world. I don't know if there was a power issue, but now you can. Yes, yes, there is an extension. I gave an extension for it. It was due on the 1st, and now we missed 2 classes, so I just pushed it 2 classes back. So you guys got an extension of 2 class periods. Okay, so let's get to the lesson today. So let me stop sharing and let's get to the lesson today. I would have come in with a quiz today, but I didn't think it was fair to come in hardcore with a quiz after we've been gone for a while. So I'm going to be really cool just to kind of recapture what it was we were doing before we left. I'm going to explain some of this. And these are the topics. The quiz for next class. I'll give you guys a moment to go ahead and write this down. You're going to need to know the rules for base pairing. So we just did some of that a moment ago. So you need to know what pairs with what. That's pretty easy. Adenine, guanine, cytosine, guanine, cytosine, adenine, thymine. But also, how many hydrogen bonds between each of those? You need to know about the structure of DNA, the subunits of a nucleotide, what the do now was. We just went over that. This Mendelssohn-Stahl experiment, I'm going to explain that experiment today. That is going to be on, your quiz next class. And you need to know all of the enzymes involved. And we're going to go over that today. It's going to be something that you cannot guess your way through. You will not be able to be like, oh, I think I'm going to shoot from the hip. It's going to be a lot of open answer. And not only that it's going to be a lot of open answer, you're not going to be able to just Google the answer. It will be situational. So there's no messing around with this. You're going to have to actually know this before. So I have hovered here for a moment. Luckily for you, I'm recording this. And if you didn't write this down, you can come back and you can watch the video to know what the topics are going to be for the quiz. And the quiz is going to be very beginning of the next class. All right, so we're going to practice some things. Also, I have built in some text. So this is going to be some text, right? There's not a lot of explaining here. This is just what it is. I'm going to go to, as I go through this lesson today, instead of timer for me, 20 minutes. That's all I get. When I go through this lesson today, I'm going to bypass a lot of the text because you can come back and you can open this PowerPoint. Right now, you can't open the PowerPoint. I'll publish it in a moment. But for the sake of time, I'd rather explain the figures than sit down and read to you from a PowerPoint. Nobody wants to be read to. So I'm going to go in here and I'm going to explain a few things. So in replication, there is going to be something called a template. We're going to be using a template. So this is going to be what we consider to be a parental strand. So think of this. This is the parent, the original strand. This is the parent, and the parent is going to leave two templates. These are going to be my templates. Now, a template or a template is going to be something that you use to copy, right? So you can see that's this here is right here. This is right here. The parent strand, you're going to break these hydrogen bonds, and I'll show you the enzyme that does that in a moment. And you just use this as a template to make the next copy. So this is the parental strand. These are the templates from the parental strand. There's two templates. And these are going to be my, these are my formed daughter strands. These are our daughter strands. The daughter strands have the template. So I've got my template from a parent, and I've got my other template, and I'll move you guys' faces, from the parent. So the parent is no longer exists, right? The parent no longer exists. These daughters are now going to be, go on to be their own parent strands whenever they replicate. So we have this, but if you'll look at it, this is an exact copy of these two. We have new strands from the template. There, I've got those templates that are still here from the parental strand, but we have the copies that have come from this. It's not too difficult of a concept. So let's practice this just for a second. So let's go, Diego. What should we put here with my base pairs? So this is my template. This will be my template here. So Diego, tell me what would be the first base. I'm going for Diego. I'm going to be Diego. Yes, there will be definitely a quiz question about this. I can't wait, so I'm jumping to somebody else. I just cannot wait for this. It's going to take too long. You've got to get through this, and you're going to have to help me out. Oh, you got it, baby. Well, thank you. Oh, it would not. So thymine doesn't pair with thymine. Thymine pairs with adenine. But we're missing something. Should be here. What number should be here? There you go. Thank you. 3. How many hydrogen bonds between adenine and thymine? How many hydrogen bonds between adenine and thymine? And there, you're correct. A lot of you guys are giving that to me. Thank you. There are 2. So I'm going to go ahead and put 2 hydrogen bonds. So adenine is going to pair with thymine here. Obviously, that's going to be 2. They're the inverse of each other. And then cytosine will have 3 bonds, but what cytosine is going to pair with guanine. Thank you for getting us started. And then guanine is going to pair with cytosine. Guanine is going to pair with cytosine. Adenine, 2 hydrogen bonds pairs with thymine. Thymine, 2 hydrogen bonds pairs with adenine. And then adenine, 2 hydrogen bonds pairs with thymine. The opposite of each other. So I've got a 5 end and a 3 end. That's it. Now, I don't have the backbone. I don't have my phosphate groups and my deoxyribose here. I'll go ahead and draw that backbone on here. But they're not asking about that. Right here, all we're talking about is a corresponding sequence, the sequence of nitrogen bases, nitrogenous bases. And that's essentially how it works. Now, based on that, which is the appropriate model? I've got 3 models. I've got conservative model, the semi-conservative model, and this dispersive model. And I'll wait for you guys 1 minute. Which is going to be the correct model? A, B, or C? Don't answer out loud. I'll call on one of you in a moment. And please don't answer it out to everybody where everybody can see it. Please don't do that. Yes, I've had a couple of you guys say, yes, this is from the reading. I know it's from the reading. I'm just reviewing what you already read. "20 seconds. Giving you time to read it. How much time do I have? Roger. Okay, drop it in the chat. Just drop it in the chat. I won't call anybody out. Just drop it in the chat. I just want to see what everybody got. Privately, if you don't mind. It's all right. Good, good. You know what, Amarani, I was going to call on you and I decided I wasn't. Now I wish I had, but you're correct, ma'am. You're correct, ma'am. It is the semi-conservative model. It is B. B is the correct answer. You can see I've got my template. This is the parental strand. And Is got a template here. I've got a template here. And now I've got daughters, right? Now here you can see my daughter. This is my parent. Here's my templates for my daughters. These are going to be my daughter strands. And these daughter strands are going to now be parents to these. So this is a template that is now going to be over here. And that original template is going to be over there. And so you're going to see that you're going to, it's just going to disperse, not disperse, it's going to keep this original strand. And you know what's really funny? I wonder where this original sequence is in my body right now. From the egg that I fertilized egg, I wonder where these original strands in my body are. They're somewhere, unless that cell has died. Those cells have died. But that original sequence that was me, I wonder where those strands are. I've always wondered that. I don't wonder it too much, but it's kind of a cool thought to think about. Okay, so we know this. Okay, how do we know this? Now, I'm colorblind. There is an experiment, and I told you guys, you will have to understand this experiment and talk about this experiment in the quiz. So this experiment's really straightforward. What they did, these two scientists, Menselson and Stahl did, was they grew bacteria in nitrogen. Remember my nitrogenous bases. So we've got phosphate group, I've got my deoxyribose sugar, and then I have a phosphate. So this nitrogenous base. This is my nitrogenous base. They grew these bacteria in nitrogen that was a isotope, a heavy isotope. So it had an atomic number of 15. And they then grew the same bacteria that had been growing in this heavy isotope. So their nitrogen is going to be nitrogen 15. They're going to be in nitrogen 15, but then they grew them in some after that were in nitrogen 14. So what they got was the first generation, after they had replicated, they could see a beautiful band right there. And then after they replicated the second time, they could see a heavy band and a light band right here. I'm colorblind, but there should be a light band right there. Is there? Okay, because I'm colorblind, I can't. This should be blue, and this should be a dark blue or purple. Is that how it is or what? It's a dark blue. Okay, so this would be a light blue, and this is going to be a dark blue. And that told them that it was going to be semi-conservative. And I'll show you the reason why. So if we could jump, you'll see when they did this, again, just like we saw before, that we had these banding. This is our dark blue band, our dark blue. And then you had the normal band. This is our light blue. So this would be the nitrogen 14, and this is going to be in my nitrogen 15s. Now, the nitrogen 15, you can see right here, this band had the nitrogen 15 in it. The light band had, that they're accompanied would be nitrogen 14 and 15 together. Nevertheless, that heavier isotope sank deeper in that test tube. You can see it right there. And then we had this. So you would consider this to be kind of a hybrid band, and this to be, these are my daughters. These are my daughters from these guys, right? So this is this template here, this template here, one over here, and then we use those two. But they were growing in nitrogen 14, the normal isotope, and it worked. Had it been a dispersive model, all of them would have been hybrid. Had it been A conservative model, you would have had a really heavy band. Remember, this is a hybrid band, so it's suspended in the middle. You would have had a real heavy band that was at the bottom, and then you would have had light bands at the top. That's not what they got. They got hybrid bands, and so they knew that this was a semi-conservative model. And no one really knew how they copied. This is prior to having really good technology, and they didn't know how this actually worked. I'm going to go past this. I'm going to explain. This is me explaining this. So I'm going to explain this. So my question is now, we're a eukaryote. If we're not a eukaryote, what would be the other type of life? There you go, prokaryote. So which is going to be simpler? I'm sorry? Yeah, which one's more simple? Yep, these are going to be simple. And since these are simple, they're going to have small genomes, really small. We're eukaryotes. We're going to have a large genome. I'm going to put large genome. So if you're going to make a copy of something and it's large, it kind of helps to do it fast. So how could I do it fast? Well, I could start different places in the genome. Instead of starting in one location and going different places, I can make what we call replication bubbles. And I make multiple bubbles. Think of it kind of like a train. So let me erase this. Think of it like a train. And if I have a train, and let's say we're going to be in Dallas, and this is going to be in Fort Worth. And all we're doing is timing how long it takes to make up that distance. So how long does it take? Let's say this takes 30 minutes for that train to make that journey. Well, what if all my objective is just to span the track? If I leave from Fort Worth and I head towards Dallas, and I leave from Dallas and head towards Fort Worth, where would they meet? How long would this take? Hypothetically, instead of it taking 30 minutes for this distance to be traveled, how long would it take for this distance to be traveled now? Let's see what we get in the chat. You are correct. You are correct. Thank you very much. It would take 15 minutes. So all we're worried about is just how long it takes to cover this distance, not whether people get to one location or the next, just how long does it take to get there. So what you do is you get a replication bubble and you meet in the middle. I'll start walking my way, you start walking yours. We meet in the middle. And that's how it was when I had a little girlfriend in high school. She'd sneak out of her house, I'd sneak out of mine, we'd meet somewhere in the middle. That way it wouldn't take 30 minutes to get there, it'd take 15. And then I could sneak a little handhold and then sneak over back to my house in the middle of the night. We would meet in the middle of our two houses so that it didn't take that long to get there. And that was our method, because maybe we only had a 30-minute window. Oh, I'm lying to you. Of course I didn't do that in high school. Of course I didn't. Actually, I think that was middle school. High school, I could do it. Yeah. Now the truth comes out. So it just makes it fast. Eukaryotes, we have large genomes, and so we do it fast. You can see a replication bubble here. You can see these bubbles right there. It's really cool. Here, and then here's another one. So if I'm doing this in multiple locations, it doesn't take as long to replicate the DNA. I'm going to go past this. This is elongation. I need to make sure I do this. Again, everything I'm passing up, I'm going to explain to you. So here we have where it gets a little complicated. It's actually really easy. It just gets a little minutia based. How much time do I have? It's perfect. You guys are going to be doing practice work in 6 minutes. So a couple of things to remember here. DNA is going to work from a 5' to a 3'. Again, here's my 3, here's my 5. Here's my 3, here's my 5, here's my 3. Now, imagine this is like a zipper. You're going to open the DNA. Now, here, let's consider this step 1, and this is going to be a little later. This is going to be time. This is going to be time. I'm going to be over time. This is the same process. And this is just going to be a longer period of time. So step 1, step 2, sort of. Now, what is cool about this is you have different enzymes. One of them is called DNA polymerase 3. And that's that enzyme right here. It's just this enzyme. And what it does is it will come in and just zip up behind it. And it's working from this 5' to what will create a new 3N. This is really the new 3N at this point, but this is going to keep coming in behind. As this unzips, this is being unzipped right here. It's just going to follow in behind it and add nucleotides, add nucleotides, add nucleotides. As it goes, it's just going to add nucleotides. This creates a huge problem for this other side. And this other side here is called my lagging strand. Lagging. What does lagging mean? If like on the internet, if the internet is lagging, what does lagging mean? I'm sorry? What does it mean, Caleb? It means slow. It means slow. This is a slower process. It is a more complicated process, so it's a slower process. This is my lagging strand. And the reason is very, very straightforward. It's not too hard. And you'll see here, it seems like we're breaking dogma for a minute. We're going from 3 to 5. That doesn't make sense. But it does when you consider that this is always working, again, towards the 3N. So this enzyme is coming in and filling in these gaps. There's little gaps here, little gaps. What's going to happen is as this opens, another enzyme is going to have to come in here and add nucleotides. And so it does it in sections. It does it in these little sections. When it hits this end, it's got this enzyme's got to drop off and come back over here. So as an example, this enzyme here, this is DNA polymerase 3, it did come in here and fill in this gap. When it filled in this gap, it dropped off and comes in here and it'll fill in this gap. And then after it fills in this gap, it's going to come in here, bind here, and fill in this gap. It does leave little holes. You'll see it's got these little holes. That's what ligase does. Ligase fills in this little bitty gap. There's a little gap here that it fills in. It's going to come in here and there's a little gap, a little bond that it has to come in here, and it adds a phosphate group and seals the deal, so to speak. But the thing that you really need to remember is that you're going to be going from 5 to 3, and this is technically going from a 5 end here. This would be a 5 end working to a 3, but it does it in little gaps. So it kind of does it in a 3 to 5 fashion because it does a gap, and then another gap, and it fills it in, but it takes longer. This is simple. All my DNA polymerase is doing is just traveling, just following. This one is doing it in little sections. All right, let me show you a different way to look at this. So this is only the lagging strand, only the lagging strand. So I'm going to start. Now, how many of you guys have used a lawnmower at your house? You mow your yard. How many of you guys mow your yard yourself? Anybody mow your yard? You do? Okay, so I've got Abel here, and it looks like there's 4 other kids in here in the chat box, or 3 others that mow their yard too. Or actually more. Okay, Gavin does, Monis does, George does, LeVar does. A lot of people do. All right, Dev does. When you start, before you pull the cord, what do you have to do first? Abel. I'm asking Abel. That's right, you got to push the bubble. Do you know what that bubble's called? You got to push it 10 times. You know what that bubble's called? It does have, you're pumping gas into it, but what is it called? You don't know what it's called? Any of you guys know what it's called? It's called, if you mow your yard at home, you've got a primer. It's called a primer. That's supposed to be, it's called a primer. You got to pump the little bulb. You got to get it started, right? So this is an enzyme called primase, and it puts an RNA primer here. This red thing is an RNA primer. And what that primer does is it gives DNA polymerase 3, this is the enzyme, DNA polymerase 3, it gives it a place to bind. It has to have a primer there to start. So it grabs that primer and then starts to go to work. Again, just like I showed before, once you get to the end, it goes backwards, but it needs another primer. There would be another primer here. There would be another primer. And it just repeats. There's not another primer shown right? The little thing, then there's gonna be another primer over here. It's gonna come back and it's gonna grab another primer and work its way there. That's my cue. All right. Once you get to this primer, notice there's a little gap. There's a little gap there. We've got to correct that gap. And that's what ligase does. Ligase fills the gap. And I'm going to fills this gap. There's my primer. I'm adding these. I'm adding to the 3. I'm always working from 5 to 3. Always working from 5 to 3. And then a primer is, my ligase is going to fill in that little gap. So I'm going to give you guys some time to work this out yourself. You might want to take a look at this really hard because you're about to actually put this together yourself. I'm going to go back to Schoology. It's where everybody wants to be. You guys can see Schoology? Okay, so I'm going to go back to class. I'm going to open up today's lesson, replication, and you have a puzzle. It's a replication puzzle. It's the same thing I just had. And you guys are going to have to share your screen. Let me make sure you can share a screen. You can, and put together this puzzle. I've got it there, and it's out of order. All those steps are out of order, and you have to know these steps. So you're going to have to click and drag these into the correct order. This will be a good way for you guys to make sure you know this for the quiz on Friday. So I'm going to have some rooms, breakout rooms. Create, open, and go do it. Put these in order. Put these in order. Just decide who's going to open theirs. It doesn't matter. Anybody can open them. You guys, let's see, how much time do I have till we get out of class? 9:40, correct? Yep. We're going to have to do 15 minutes. That's all we can spare. You guys can probably do it really fast. But John, one more time.
Did she say it?
What up? So I'll come in here. Yeah, you could. I would. I would share the doc with everybody if I were you guys, and you can come in here and make your own tonight. But I would share the document. But I would sit there and talk our way through it. I would look at the document with somebody and talk your way through it. You know what you could do if you wanted to, if it's giving you a fit, is just download it as a PowerPoint and use it as a PowerPoint. But let me see.
Not. You said it was like stop motion. You tried doing it. You have a whole.
We got
you
10
have a
minutes.
whole setup to school, bro. Here you go. Here you go. Here you go. Notes. Oh no! This is just this. Oh no! Wait. I need to go back and look.
Are you guys looking at the same document? I don't mean, it's hard to know whenever I don't see you guys sharing your screen anymore.
Oh my gosh. Okay, I'll just. What happened?
Okay. Hey, are you guys using the same document? I don't see anybody sharing their screen.
Yeah, Keaton shared his copy with the rest of the group.
So how do you guys know? Thank you, Keaton. How do you guys know that what each other are doing? I mean, if you're not talking your way through it, maybe you are.
I mean, we can see the live editing in the document.
Okay, yeah, I suppose you could do that. Yeah, one of those two is correct. I don't know which one. Are you guys stuck?
I'm having a technical error with Google Docs right now.
One group downloaded it as a PowerPoint and shared their screen and used PowerPoint and it worked better.
Yeah, that might be smart.
If the problem is going to be if you use a PowerPoint, you won't be able to share it between groups. You'll have to share your screen if you're using the PowerPoint to put it together. I'm going to jump to another group and check on them. Great to see these people. Randy, how are you guys doing on a scale of 1 to 10? Not great. All right, let me go over there.
Who said no?
So it looks like you guys have step 1. I don't know if I'm muted. Yeah. You got step 1 for sure. Let me look at step 2. DNA polymerase 3 adds DNA nucleotides to the primer. Yes, you got step 2. Let's look at step 3. Yes, you got step 3.
Oh,
I need to expand this so I can see it better. It's hard to see. Let's see, DNA after DNA. Okay, and it falls off. DNA polymerase 1 replaces the RNA primer, adding DNA. Yes.
I am.
I think yeah, it looks like, yeah, it looks like you guys did great, Randy. Why did you think that? Okay, yeah, you guys got this. Y'all the boss.
Yes. Yeah, we can see it.
It's supposed to be like up here.
Having technical issues.
Yeah, just hang on. I need to move your face.
Huh?
His face was in the way, and I couldn't. Oh my gosh, I can't grab the thing.
Don't worry about the doc as much as the text, man.
It's like right over here. That's the ligase that forms a bond. If I can just move this one out of the way and put this one down here for a quick second. What, I'm gonna put this back down here. Ah! Can I grab this? My God. Stay up there then. So these faces to the end of fragment 2. So this one would be, and this is correct right here. This is correct because it's ending to the end of fragment 2. So this is the 5 end, and it's adding the 3 end. Then this one would be it right here.
What's the difference between 5 and 3 end?
Well, 5 end is where you begin with. That's how you start replicating the DNA.
Okay.
And the 3, this would be the 3 end right here. This would be the 5 end, because we always start with the 5 end. But since we're doing the lagging strand, we have to somewhat start in by sections
Oh, I see.
or pieces. So this is what the primer. This is primer, and this would be the 3 end right here. Since it's going this way, that means the end right here would be the 3 end, and this would be the 5 end.
Okay.
So this one would be correct right here. This one, whole 3 adds to forming Ozaki fragment.
Yes, hey, don't go back.
Go back.
That's correct. So that where it says DNA polymerase adds DNA nucleotides to primer, that is that second image. You're correct
one
on that. Yeah.
This one here.
B, that corrects. But look where it says DNA polymerase 1 replaces RNA with DNA. So where is that happening? RNA is the red part.
RNA is the red part. You mean this
Primer.
right here?
Yeah, that's
The-
RNA primer. So that where it says DNA polymerase 1 replaces RNA with DNA. Do you see that in that second little image? You don't. You're looking for where the red is going to be going away.
Oh, so it would be this one right here.
Oh, there you go. I've got to bring everybody back. I had 2 groups that already finished. I don't know how many have got it now, but this will give you guys, I would meet and try to work on this
Gosh,
evening.
damn.
Yeah, I was doing that for me.
Yeah, it's
You guys can stay until it kicks you out completely.
Okay.
All right, do me a favor and drop it in the chat. I know two groups did finish it, but I was with another group at the end. I don't know how many groups finished now. Did 3 or 4 groups finish? Just drop it in the chat. Did you or did you not finish? Did you or did you not finish? Okay, so it stayed the same. It was 2 groups. No, apparently 3. Yeah, 3 of you guys did finish. So it was a little mixed. Some of you guys had some technical problems, but hopefully you guys were getting the point. I've got the solution here, and I'm going to publish this so you guys can see it later. The thing that you really want to focus on when you're looking at this and trying to, and you do have to memorize this. It's really not hard to memorize. Primase is going to be dealing with primers. My primer is going to be this red part. It's RNA, not DNA. But if you read each description and then try to match it to these figures, or at least these different steps, you can see how you will give you some time to actually think about this and marinate on this as you go. So let's see, there's a chat. Yeah, it is. It should be in your book as well. I mean, if it doesn't have Ozaki fragments, then your book's terrible. And I did put the pages for this. It's the pages that you guys have already read. If you've got the book from the school, it would have been the pages you guys have already read. This is just it in a lot more detail. All right, I need to finish, and then I'm going to release you guys to your homework and show you what your homework is. Hopefully I can finish. There we go. So on this slide, you must know these. I'm going to release this PowerPoint so that you guys can come back and review it, but you do need to know these. So on an open response, you need to know what these different enzymes do. What you guys were just doing was using these different enzymes. The only enzyme up here that you really have to know is this one. Helicase is going to be what unwinds or unzips makes the replication fork, what makes that split. So if this is this is going to be, imagine an enzyme that's unzipping the zipper. That's what helicase does. Helicase comes in and breaks those hydrogen bonds. Remember, they're very weak, so it's easy to do. It just comes in here and unzips it, just like a zipper. These 4 enzymes are what you were just using. Primase gets it started. DNA polymerase 3 adds nitrogen or bases, nitrogenous bases. DNA polymerase 1 removes the primer, adds DNA, and then ligase fills in the gaps, removes those gaps, fills it in. It joins the fragments together. It's not hard to do, but it does take time. You're going to have to come in and study it to get this. This is it in detail, what I was just showing you before. What you just saw, this image is all of it. And you can see it's working in different directions. So the Ozagi fragments are up here or on top. And here you've got Ozagi fragments are going to be here on the bottom. This is going to be the leading strand. You can see it's continuous, but the fragments are going to be what's coming in as the gaps or in these bubbles are built. It's pretty elegant, to be honest with you. Last thing I have to bring you in to explain to you is something called a telomere. Let me get to this. So we do have, this is just the ligase. This is it, me coming in and bringing in this last little part. But you do have something that's called a problem with replication. Every time you go through replication, your DNA is going to get shorter. It's not going to, your DNA, you literally lose DNA every time you replicate it. You lose, so if this is my chromosome, imagine this chromosome that at first replication, the first time at replication, let's say that this is the 50,000th time you've replicated it. It's going to be not as long. It's going to be, so let's say if we're adding, I don't know what units are. Let's say this is 100 arbitrary units. We'll call these arbitrary units. So just whatever units, doesn't matter. I don't have any units, but let's say it's 100 whatever units long. After 50,000 replications, you may be 80 of those units long. That's a real problem. Every time you do this, you're going to cut out DNA. And remember, your DNA has sections in it. And what are those sections that code for proteins? What are those sections in your DNA that code for proteins? What do we call those sections? What do we call the sections that code for proteins? It's an open question to anybody in the group. What are those sections that we code for proteins? And you've got a bunch of them. So let's say this is 1, this is 2, this is 3. They're little sections of DNA that code for proteins, that give you your traits. Ah, thank you, Santos. Could you text that out to everybody? But that, you got it, sweetheart. They are genes. Well, what happens if now I'm in my first unit, so this is right here. Now I don't have all of those. So now let's say I start at 2, 3, 4, 5, but I'm missing 6. I've got part of 6-ish, and 1-ish. If I lose those genes, if I start cutting into those genes, then I'm going to stop producing those proteins or start producing those traits. This is something that you're probably more familiar with than you realize. At the end of every one of your chromosomes, you have these little things called telomeres. And I've got a good analogy for what a telomere is. They're basically, think of them like tread. A telomere are these little bitty extra sequences at the ends of your chromosomes. They're little bitty sections, and they're expendable. They're really expendable. They don't code for genes. It's just little extra pieces of DNA. Think of it like the tread. This is a lot of telomeres, so many telomeres, or long, let's not call them many, let's call them long. These are long telomeres. Telomeres. So over time, though, as you replicate, these are going to be short telomeres. You cut into them. Every time you replicate your DNA, you cut into your telomeres. Eventually, what happens when you don't have any more telomeres? What happens? Well, what happens with the tire? This is tread, right? This is tread. You've got tread on your tires, and this is very little tread. Very little tread. What happens after you get into your tread? after you have got your tread, you've been driving on it for a while, and now, oh, you start to lose grip, you start to lose genes. Yes, you'll start to cut into like the in this case, you guys may have seen this. There's like metal in here. There's parts of the tire that keep it really structurally sound. This is all sacrificial. You can sacrifice your tread. You can sacrifice it for a while. This is sacrificial, but then you get into the metal. The metal, think about this as being the genes. This is the structure of the tire that's really important that you have to have. Yeah, you're going to be in trouble. So this is a young chromosome. This is an old chromosome. This is a young chromosome. This is an older chromosome. All this is time. This is time. This is telomeres. Telomeres intact. You have a lot of tread, and this is telomere loss. You no longer have those telomeres. You're cutting, cutting into genes. You're cutting into your genes here. So like this old man or old woman, they're losing genes that produce proteins that make their skin flexible, or they produce the oils that they need in their skin. They're starting to lose these genes. And if you start cutting into your genes, you're going to start having some problems. Your skin no longer functions like it used to. This is an issue. It could have been that's what was happening to some of my skin and why I got the cancer. I don't really know for sure. But there's going to be there's issues here. So these little tips of these chromosomes, you can see they're pretty small. They're pretty small. They're little. They're not designed to last forever. There is a finite amount of time that you're going to be on Earth, and a lot of that is going to be dictated just into this. Once you start cutting into this, you start to lose genes, you start to lose traits, you start to lose traits, you start to lose function, you start to lose function, you lose your life. And so there's nothing you can do about this unless you can put some caps on them. There are some organisms that have little caps on these, and we're not one of those organisms. If we were designed perfectly, this would not happen, but we are not. Let me show your homework. Oh, we got some chats here. Yeah, they don't work anywhere. They're trash. Yes, you are losing part of, yes, Gavin. So yes, so every time you replicate, you're losing part of your DNA. You're losing the little tips and the ends. It's just getting shorter and shorter and shorter and shorter and shorter and shorter and shorter. Eventually, it no longer works. All right, so let me get back to your page. I'm going to do a couple things. I want to make sure that you could see it. That's not the right location. Yeah, you can see it. She can see it. I'm looking at Randy's to see if she can see. All right, where is your homework? Somebody asked. It is right here. It's called Replication Notes. Now, please pay attention and see where it says these will help you on your quiz. It says this will assist you with the quiz. It's not going to solve the quiz for you. You're going to have to study it. I would finish this puzzle, look at my replication notes, and I've got information from our previous lessons that are in there as well. So I know it's been, I would have given you guys a quiz today had we had the next class, but that wasn't fair. So hopefully you've been studying. These are your notes. I'm not going to give you a grade for it. I'm going to look to see if you did it. And if you didn't do it, heaven help you, because you're going to need some spiritual, supernatural help. It's going to take that. But the one thing I really want to emphasize is when you go on here, you'll see that I have a part. Oh my God, it's not going to let me show it to you. I don't know why. Why won't it let me show it? It's weird. Maybe. Hey, Jacob, will you open your document and let me see if it'll let me see your document? If you do that, just open it real quick. I want to point something out. Just open your document. Just open it real quick.
I'll refresh my page.
You've already opened yours? Thank you, sweetheart.
Let's see
Oh, it's
where.
not working for me.
Huh, it's weird. Maybe it's just being strange. Let me get. Randy, I can't see yours either. It may be because I have not logged into my Google account. No, I'm logged in. Okay, here it is. Here's Randy's. Randy, can you did it pull up this same document? Everybody can see it. The last part is going to be really important. And I built this for you guys so that you could do well on this quiz. If you are at home and you'll see, you can't obviously draw on this document. You need to draw this like, obviously on a piece of paper at home. You can do everything else on here, but at home, by drawing this, it's gonna help you remember it. It's gonna help you remember it. Label this, it was basically what we were doing earlier, but drawing it on a piece of paper. And by doing so, you're going to remember it more than if you were just to be looking at it on a PowerPoint. Make that drawing. There's another reason. On the exams, when we have the exam at the end of next year, they're going to ask you to make these drawings. You literally have to draw on this test. It's very different than AP. You have to make these drawings. So this is extremely good practice. It will help you out a lot on this. And then finally, go back and just do the reading. The reading is, it's only 5 pages. It's going to help you on this class, and it's going to help you on the next lesson as well. All right, your exit ticket for today is just to drop me a chat, and privately, I want to know on a scale of 1 to 10, how well did I do explaining this? 1 to 10. And then you can leave, but I will also stay and answer any questions for the next 5 minutes. But if you want to leave, you can leave, but privately chat 1 to 10. 10 being awesome, 1 meaning I suck terrible. And I'm not going to take offense to it, and I'll stay and answer any question. Oh my God, what was that? Peerings and pyrimidines, that's just the guanine, cytosine, adenine, thymine. From the front office. At the school. No, downstairs. Yeah, go to Patty. Go to Patty. Patty's got it. I'm open for questions. You guys have any questions, just go ahead and shout them out. Yes, I have office hours today. Yes, I have office hours today. If you want to come, I have office hours. I don't know, Janiyah. I don't know, sweetheart. I would call the front office and ask Patty. She has copies. She just might have to go to the book room. But yeah, I mean, I know she still has books. So Jacob, if your DNA replication notes aren't working, ask somebody to share a copy with you. And if not, you send me an email and I'll email you a copy. Yes, the quiz is going to be tough. I promise. Somebody asked about that. Yeah, the quiz is going to be tough. I'm going to have to ramp up the rigor for the rest of the year. I'm here to answer any questions that you guys have. If you have questions, you can, I'm staying back. Otherwise, you don't have to turn in the replication puzzle. There's nothing that you have to turn in. Yep, you got to clean your desk. That is it. But what I would do, I'd probably look online and find a find an image there too, to look at those. And just, if you draw it, for me, I always drew everything that I was learning in biology because it helped me remember it. Like I would read it, I would talk to my study group about it, I would draw those images. There were no, we didn't have PowerPoint. Like when I was in college, everything that, like whenever I had a professor teaching us this, it was just them up here going like this, dot, 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 no pictures. The only picture I had was in that book. So I was like, I would have to go home and just draw and study. There was no PowerPoint or anything like that. Just a guy talking. It was crazy. It was crazy.